Feel like spring or summer? Feels like spring? It's nice weather. Thank you for coming to lecture inside. I know it's tough this time of year when the weather gets really nice to, stay the course. But we really only have about 5 weeks left. Okay, or less. Everybody have a nice Easter weekend? Anybody do anything fun? Did you go home? Eat to your heart's content? No? Yes? No? Anybody get outside? Yeah, it was a nice weekend to be outside. Yeah, there's still 10 feet of snow at Snowball. Yeah. Pretty amazing. Okay. Couple of announcements. Everybody get the reminder on Sunday, Easter Sunday, about the quizzes? I didn't want that to... I know it was kind of a holiday weekend, so I didn't want you guys to forget that. So the next quiz is due, if you didn't already do it, some of you already did it, I think, quiz 12, but the next quiz 12 is due this coming Sunday by 11:59 p.m. Doesn't mean you have to wait till Sunday. You could do it anytime this week. Probably not a bad idea because they're pretty easy questions, and you can look up the answers for crying out loud, right? And it's going to get you in the book and have you look at figures like this. Okay, a perspective. Again, thanks for coming to lecture. I know it's gorgeous outside and you got to be in a classroom and you listen to me for an hour and 15 minutes. But how many of you played or play a sport or an instrument or have something that you're passionate about where you need to practice? Okay. Nobody's passionate. Some of you aren't passionate about anything, huh? Okay. All right, fair, that's fair. Definitely not this class, Keller. Not passionate about this class. Well, we're trying to change that. So if you've got a sport, an event, how often do you practice to get good? Every day? 2-a-days? Anybody do 2-a-days when it's getting really important? Okay. If it's athletics, 2-a-days, you can only do that for so long, and your body tells you it's time to take a break. Okay. Maybe every day, maybe 3 times a week. Hey, maybe think back to high school, maybe played a sport or had an activity. You're in a club, even student council. How often did you meet? Regularly, we'll say that. To get better at it. So it's beautiful outside. You're in here because you're committed to this next exam. And the only way to get better at it isn't practicing the day before the football game. Your coach didn't tell you, hey, game on Friday, we're not doing anything till Thursday after school. I'll see you then. Is that what he said? Your coach of your basketball team didn't say, hey, we've got a girls' volleyball match or a girls' basketball match on Thursday evening. We're going to practice Wednesday. Just don't do anything until Wednesday, and then I'll see you on Wednesday. So in athletics or in our club activities or in our, passions that we have, whatever it is, you practice regularly. And many of you have told me in office hours, bless you, that your plan, your long-term goal is to go to PT school, nursing school, med school, dental hygiene. The weekend before the exam, the night before the exam, isn't the time to study. It's the time to maybe study and review the things that you've been doing, maybe not daily, but at least twice a week. Here, I'd like to believe it's more than just twice a week. Does that make sense? I'm trying to put it in perspective of, this class is going to take some energy. And if you're willing to put in the energy, you'll see the outcome on exam day or game day, match day, the day of the competition. And that's what we're working towards. Okay? So I'm really proud of all of you for being here because it's beautiful outside. But that's the kind of level of commitment that it takes. Right? Some of you that practice outdoors in Phoenix in August, for your sport, you didn't want to be there, but you were there because it made the difference, and it made you a better athlete or performer, whether it's dance or a musical instrument, right? Nobody just practices the piano the night before the rehearsal or the recital and nails it. I mean, you practice on a regular basis. So that's what I'm trying to get you all to convert to, is practicing regularly. Okay? Make sense? And that's your future. If you're going to lean into those professions, that's what's going to be coming. It's not going to be once a week. PT school, PA school, med school, it will be likely daily, like somebody said. So I want to frame it and have a realistic expectation. Okay, this is where we left off, but before we go there, why are parallel lines so lonely? Because they never meet. Well done. What did one elevator say to the other? You're going down? I think I'm coming down with something. How's that? Some of you have colds. You're getting over the sniffles. What's a frog's favorite kind of flower? I thought spring. This made a lot of sense. Frog's favorite kind of flower. What's that? A lily pad? I like that one. Maybe an alternative would be a crocus. Okay. All right. So best I got. I don't know if any of you are watching the HBO show with the fungus. Anybody watch that series? Yeah, Last of Us. I love the dad joke book. I mean, come on, you guys, that's just awesome. No? Okay, all right. Well, I'm not as grumpy as that guy. He laughs sometimes. All right. Here we go. You ready? We're talking about muscle physiology, but first we got to get into the anatomy. And I want to remind you that the structure of the muscle cell, otherwise known as what? What's another name for a muscle cell? A muscle fiber. Thank you. I heard it from a couple different locations. That tells me from last Wednesday you were looking at the material, which is great. A muscle fiber, a.k.a. a muscle cell. The way that cell is organized, its anatomy, its structure, is going to allow for a very specific type of function of that cell. And if you can see that motif right from the beginning at how goofy this cell looks, okay, you're like, that is some jacked up cell, Keller. It is extremely specialized, which is what makes it so cool. But there's a lot of components inside of this cell, otherwise known as a fiber, that you all know. This is Unit 1 review. You're like, I hate when you say that. But it is. This is Unit 1 stuff coming back. And if we didn't talk about Unit 1 stuff to remind you of the parts of a cell, when we get here and I start talking about mitochondria content, or I start talking about a modified sarcoplasmic reticulum, okay? Or I talk about a modified Golgi apparatus, which is a sarcoplasmic reticulum in a muscle cell. You start seeing that there are what we call homologous parts. There are parts that are very similar from a regular run-of-the-mill cell that's kind of boring to a very specialized cell. But if we don't point back to Unit 1 and you don't have that material as a foundation, I got to go way back into high school biology or basic bio that you didn't have for me, or maybe you didn't remember it. And so at least we have this grounding point. And everything that we're going to point to today should have that framework of, okay, I think I've seen this before, it just looks differently. So the parts, some of your SI materials this week are going to be asking you to label this structure just that you have here. Very relevant stuff, because if you dive down into the basic anatomy, we package these filaments that we're going to look at, actin and myosin filaments, or the thick and the thin filament. We're going to look at these filaments, and we're going to move and zoom in into the microanatomy. But you need to know where you are. Now, these filaments all bundle together, and they make up the architecture of the inside of the cell, which is the myofiber, or the muscle cell. And then you can sort of appreciate that there's a sarcolemma. The sarcolemma is the membrane. It's the plasma membrane. You've got mitochondria. Well, oh my gosh, there's more than one. Is that allowed? Yeah. What does that tell you about the energy demand of a cell? It's very high. Look at all the mitochondria, which are considered the powerhouse of the cell. They generate ATP. In their cytoplasm is where glycolysis happens. Right? We also see if aerobic respiration takes place within the mitochondria, in the presence of oxygen, do you get more or less ATP? You get more. Does it take longer? Is it faster or slower than glycolysis, aerobic respiration? It's slower, but you generate more ATP. Now, am I going to ask you about the numbers? No. But do I want you to have the reference of, okay, now I understand why mitochondria are important. Now I understand why there's so many here, because this must be a high energy utilizing tissue, which it is. This could be, we're talking about skeletal muscle, but if you're thinking about cardiac muscle, well, you don't want it to run out of energy in the middle of the day. That would be bad, right? And cardiac and skeletal are very similar. You'll get cardiac muscle next semester in 202. The foundation of skeletal muscle is very applicable to cardiac because they're very, very, very similar. Here's the nucleus. Here's what we call the sarcoplasmic reticulum, which is a modified Golgi apparatus. And the sarcoplasmic reticulum here has a function of storing calcium. And I just want you to pause that statement. Because we're going to come back to the role of calcium in this. And I just looked at my notes. I misspoke. Not Golgi, endoplasmic reticulum. My bad. Modified endoplasmic reticulum is the sarcoplasmic reticulum. We're going to see how it's organized very specifically around these triad units, where we've got these three tubes. And down the middle of that tube is what we call the transverse tubule. And we'll talk about the role that it plays. And then on either side of it are these terminal cisternae, which are basically the terminal tank of the sarcoplasmic reticulum, the end storage house. That's what that word literally refers to, terminal cisternae. A cistern is just a tank. Okay, so this is the end tank. And all of this architecture here, this blue spiderweb looking thing, is the sarcoplasmic reticulum. And so it's continuous with this terminal cisternae, and it allows for calcium to move out into the cell or resequestered into the terminal cisternae, which is a portion of the sarcoplasmic reticulum. This is all microanatomy, and I'm just trying to make some parallels between a regular cell and a muscle cell. Or a muscle fiber, you can see this whole circle is around, this whole thing is the cell. This whole thing is the muscle fiber. And this is the inside as you peeled away the cell membrane, known as the sarcolemma. Does that make sense? It's important you know where you are. It's like a Google Earth zoom in now. Okay? So now we're here. We're going to take a look at these filaments, and we're going to go to this picture. And we've got, right in the middle, we've got thicker filaments, which we creatively, as scientists, refer to as the thick filament, because that's how we are. Then we have these other filaments here in red, which are thinner, so we refer to those as thin filaments. You tracking? Pretty straightforward. Then we have kind of this little spiral one that actually has a long segment. It's actually colored green. and it's attached to the thick filament. We call it the elastic filament. And another name for it in the literature is the titin molecule. And we actually have a researcher here at NAU who is actually world-renowned for her work on the titin protein. But it allows the muscle to contract and then go back to its native resting state like a rubber band. That's the elastomeric component, is what this elastic filament or this titin protein does for you. We've got all these different regions that we're going to describe, and I've got some electron microscopy that's pseudo-colored so you can see different zones. But really right away you should see anchor points. You should see this is called the zigzag Z-looking structure we refer to as a Z disc. Makes sense? We don't call it the I disc, the L disc. We call it the Z disc because it looks like a Z. You with me? There's one over here. It looks like you've got your thin filament and your elastic filament attached to it. And you've got some going this direction, and you've got some going this direction. You with me? Then right down the middle, you have what's called the M line, which isn't labeled on this particular slide, but just trust me. And this is the anchor point for the thick filament. And we're going to get into this today because I asked you to look at the sliding filament model. We're going to watch here towards the end of the lecture how these thick and thin filaments slide across each other, over top of each other. And the Z disc and the Z disc actually get closer and further apart during contraction and then relaxation. You with me? And that, there's a question here, hang on. That is happening at this level, inside the muscle cell. The muscle cell itself is actually doing this. Right? It's flexing on its own, contracting. So this is the anatomy, the microanatomy. You can see the corresponding electron micrograph up top. This is a cartoon or a schematic diagram. And we'll come back to these different areas like the H band and the A band and the I band. But a sarcomere is from one Z disc to another. And if you recall, and then I'll get to your question, Matthew. If you recall, back in histology in the beginning of the semester in lab, you looked at the different types of muscle. And one of the characteristics of striated muscle, which was skeletal and cardiac, is you could see striations in the histology. And this patterning, you see how it's dark, light, dark. That's what gives striated muscle its striated appearance histologically. Okay? It's the organization at the microanatomy level. Matthew, go ahead.
I was just wanting to ask if these titin molecules and what we're looking at here is a closer look at action potential and if it's in relation to it.
How is this related to action potential? This really is unrelated to the action potential, although it's coupled to it. And we're going to talk about that. We'll probably get to that Monday. We might touch on it at the end here. It's called excitation-contraction coupling. So what we did in the last unit is we had the nerve action potential, and then it went to a synapse. And we talked about things that happen in the synapse, and that's where we stopped. We're going to go nerve, and then we talked, on occasion, nerve, synapse, nerve. You remember that? Because we talked about excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Now we're going to go nerve, synapse, muscle. Okay? And these are efferent nerves that go to the muscle. They're motor nerves or motor neurons that go to the muscle and tell the muscle to contract. So to Matthew's point, it's going to be action potential, neurotransmitter, calcium, and then all this magical stuff that happens in the cell that you haven't told us about yet, which is contraction. Excitation, contraction, coupling. So this is why we do nerve physiology and then we do muscle physiology. I don't know why so many textbooks do it not that way, because to me it makes more sense. We teach it as excitation, contraction, coupling. Okay? Do you have a question? Okay. All right. So The striations formed by the dark light patterning. And we've got these different areas in the muscle that we talked about. So the A band refers to anisotropy, is the name for it. And that's the property of being directionally dependent. So these are directionally oriented. And in these bands, we have these little like zones or regions. So we've got our A band, which is basically from one end of the thick to the other end of the thick filament. Thick filament being this purple one right here with these little globular heads that we'll look at later. And we've got now drawn here is the M line that I was telling you about. That's the anchor point for the thick filament. And the H zone or the H band is where there's only thick and there's no thin. The A band is the entire length of the thick filament. So you can see how the H band is a subset of the A band. You with me? Now the I band. The I band contains the Z disc as well as the titin molecule or the elastic filament. And so here's the I band where there is no thick and only thin. I band over here, no thick and only thin. Contains the Z disc because the thick and the elastic filament are bound to the Z disc. From one Z disc to another is what we refer to as a sarcomere, the distance between two Z discs. And a sarcomere is a Greek term. So the first sark refers to flesh, and meros is part. So it's part of the flesh. That's how it's named. And sarcomere is actually referred to as the functional unit of muscle. Functional unit of muscle. So that would be important terminology to write down under sarcomere in case you get a question on the exam that was what I refer to as a level 1 question, which is just define it. What is it? What's a sarcomere? Well, sarcomere could be defined as either a Z disc to a Z disc, or it could be defined as the functional unit of muscle. Both of those answers would be correct. And if one was labeled A and the other was labeled B, and then the third answer was part of the action potential, that's wrong. And D was A and B only, and E was B and C only, what would you choose? A and B only, right? You see how that works? And that's how you take a level 1 question and make it a level 2 question. Where instead of like high school questions of what's a sarcomere, like, why didn't you just ask that question? Because that's the kind of questions you get in high school level classes. Here, what's a sarcomere? Z disc to Z disc, functional unit of muscle. Both of those are correct. That could be an A and B only answer. You with me? Okay. So now let's take a peek at these striations in a little bit more detail. We've got myosin and actin. Bless you. The myosin is what makes up the thick filament, and actin is a major component of the thin filament. There's 2 other proteins that make up the thin filament, and we'll talk about those in a second. But actin and myosin are the 2 that interact. So there's a couple of different characters in this story. Actin and myosin are probably the main characters. Okay, if it's like a play or a movie. And then you've got some supporting actors. These are the lead actors, you have supporting actors. So we'll look at some of the supporting or what we call regulatory proteins here in a second. But actin and myosin are proteins that are in all of our cells in some capacity. In fact, in basic bio, you looked at actin as a main cytoskeletal protein. You remember that? And it keeps the cell open. It keeps it from collapsing. Actin will move. It'll actually extend and shrink as the cell goes through locomotion. Moves. So here, the strategy is let's use the same exact proteins that are in our cells and let's organize them in a very specific pattern. So they function in cell motility, mitosis, the transport of intracellular material. That's all cell and molecular biology stuff. Here, we're going to borrow them, and we're going to organize them in a precise way for both cardiac as well as skeletal muscle, which are our two striated muscle types that you learned about in lab and we talked about in Unit 1. And they overlap in this architecture. So get very familiar with this type of diagram. But these pictures that are really, I think, pretty pictures to study from really help you, I think, kind of ID when you go back and forth between this diagram and these pictures of where things are. So they're highlighted. Here we've got skeletal muscle. These are the striations. This is electron microscopy that has pseudocolorations here, where we've got the fiber highlighted in blue. We've got the A bands. It almost looks like tire tread. You with me? The A bands highlighted in blue. Here we have the I bands highlighted in blue. We're basically alternating. Here we've got the nuclei, so they're multinucleated is what skeletal muscle is. So our striations are made up of these different bands, right? We've got anisotropy for the A band. We've got part of the A band, bless you, where the thick and the thin filaments overlap, where that's especially dark. The region of the H band, that's the middle, and you've got thick filaments only. That's what we just talked about on that diagram. Thick filaments only in the H band. M line is the middle point, the anchoring point. I band is an alternating light band, right? That's the alternating light band, the I band. Stands for isotropy. It has properties in all directions, not just a single direction, like the A band. The Z disc is the anchoring point for the thin filaments and the elastic filaments. And it comes right through the middle of the I band, or bisects the I band, on either side of the sarcomere. Some more pictures. Sarcomere. You can clearly see the Z line, this dark line right down this side, and a dark line right down this side. It almost looks like a window pane on the left and a window pane on the right. Over here, the Z disc is highlighted for your convenience, so you can kind of appreciate where we are. Here's our I band. Here's our M line right down the middle. We have our A band. We have our H band, which is obviously going to be thicker than the M line, because the H band is that region where there's only thick filament that encompasses the M line. Here's a sarcomere defined from Z disc to Z disc, or Z line to Z line. And this whole structure is the myofibril. Myofibril. Okay? Z disc to Z disc, functional unit of a muscle fiber. These muscle cells shorten because their individual sarcomeres shorten. So when you want to contract, you actually bring Z disc closer to the Z disc. Is this making sense? How this works, okay? What's important to understand is the thick and the thin filaments do not change length. So is there a physical limit to how much a muscle can contract? Yes. There is a stopping point where you have this thick filament hits the Z disc. This thick filament hits the Z disc, and you basically kind of like bumper out, if you will. So it's important to note that the thick or the thin filaments do not change length. They just change in the amount of overlap over top of each other. It's like a telescoping pole, right? You ever worked with one of those, like a pool pole that kind of telescopes out? Well, there's the shortest length that pole will get, right? And its maximum length is about 2x the length of the starting pole, because you have a pole buried inside the pole, right? And then you can telescope it out, you lose a little bit because of the overlap that stays, but there's a maximum length and there's a minimum length of that pool pole. Well, there's a maximum and minimum length of the sarcomere. But the thick and the thin filaments never change their length. Just like those two telescoping poles never change their length. They just change their relationship to each other. One's buried inside the other until you telescope it out. Now when this is happening, you have to have this structure that's sliding across each other. You actually have it anchored to something else in order to generate force. You with me? Because if it's not anchored to anything outside of the cell, you'll have no force production. And that's where you've got dystrophin and other linking proteins that attach on the extracellular matrix proteins. And they use these transmembrane proteins that are embedded in the cell surface that connect the inside of the cell to the outside of the cell. Do you remember those? Those embedded transmembrane proteins that go all the way through the phospholipid bilayer? These filaments that are sliding inside the cell are anchored to one of those, which is then connected to the outside of the cell, and that's your anchor point to allow you to have a force production. So we just got done with winter. You can have the biggest, baddest engine, four-wheel drive, gnarliest tires. If you have no anchor point because you're sitting on a sheet of ice and you rev that really amazing engine, what happens? You just spin. I saw a lot of that this winter. So you have to have an anchor point, and that's what dystrophin and linking proteins do. Now, we've talked about muscular dystrophy in passing. So Duchenne's muscular dystrophy is a disorder that affects dystrophin specifically. And we'll talk about it here in a little bit. In fact, it's likely the topic of your bonus opportunity this unit. Okay. I got to get through the paper first before I can build it for you.
So if they don't change the length, do they stretch?
Nope.
So then what's the elastic?
The elastic filament is to pull back on the stretched out proteins to bring them back to resting position. Okay? No, the elastic filament. So there's a thick, thin, and elastic. Let's just go back real quick. You see right down here, this elastic filament, it's colored in green. It's really, really hard to see. Better picture probably in your book. You can look at it. Let's see if I have another picture. Here we go. This one's bigger. It's this one right down here. See how it's kind of like a corkscrew and then it has a thin line? That's your thin filament. That's your elastic filament. And it's connected here to the thick filament.
And that's what
Pulls it back to the resting position. Okay. So this concept's really important that you understand that you can generate force, but you have to generate force with an anchor point in order to translate the force to move the muscle. Otherwise, the cells are just going to be doing this all by themselves. Okay, so now we're going to introduce some of these additional characters. We're going to, in our story, if you will, about contraction. We got thick filaments, we have thin filaments, we have elastic filaments. Another name for elastic filament is titin, or the titin protein that makes up the elastic filament. If we zoom in on the thick and the thin, which is primarily what we're going to focus on, we won't spend a lot of energy talking about titin or the elastic filament. We're going to focus most of our energy on the thick filament and the thin filament. The thick filament is made up almost exclusively of myosin as a protein. And this myosin has two components. It has a sheath or a spindle, and then it has this globular head. And it kind of looks like a hammer. So it's got a long shaft with a head, and the head can actually be in different positions. And that head changes positions based upon how many phosphate molecules are on it. And the phosphate molecules are coming on or off ATP, which is the energy source. And where does the ATP come from in the muscle cell? The mitochondria. And so every single one of these heads is going to have ATP bound to it. And there's a lot of myosin heads in a muscle. So you need a lot of ATP, hence you have a lot of mitochondria. You follow? Okay. Next week we'll translate this all the way to performance with muscles. We'll get into a little bit of exercise physiology stuff, where it's like, hey, what happens if you're a sprinter? Right? What if I was a running back? What if I was a 100-meter sprinter? Right? What if I was a long-distance runner? What if I did like the 4x400? What if, right? We'll see some specialization of these muscle cells based upon the type of activity. We'll talk about that next week. Right now, we're in the weeds. We're super in the weeds. We're like inside of the cell. And we'll back it out to like application next week. Now we look at the thin filament. The thin filament is very, very different than the thick filament. In fact, one of the big differences is it's got other types of proteins. So, the red globular or circular balls or spheres are actin. And that's the main component of the thin filament. But then you can see that there's this sort of like white-colored, cylinder-looking protein known as tropomyosin. And then you have another globular colored in yellow that attaches to tropomyosin called troponin complex. So we've got 3 important proteins for you to remember architecturally on the thin filament. Actin, also referred to as G-actin. Tropomyosin, which is a long cylinder-looking like protein. And then troponin, which is a globular protein, much like actin, but in far less quantity. Troponin and tropomyosin are referred to as regulatory proteins. They actually determine if myosin is going to interact with actin. Because underneath this tropomyosin, there's a binding site for this myosin head. So the tropomyosin covers the myosin head binding site. Did you follow that? It's called tropomyosin. This is a myosin head, so maybe that's how you remember it. Tropomyosin has to move out of the way to expose the binding site for the myosin head. The binding site is located on the globular, red, spherical-looking actin protein. And if tropomyosin is blocking the binding site, the myosin head can't bind, you get no contraction. Once the tropomyosin moves out of the way, the binding site's exposed, the myosin head can find the binding site, and you have a connection, and then you're going to move that myosin head to actually slide the filaments. And we'll talk about this in more detail many, many times. So that's kind of the story. We're going to fill in more of the story, and we're going to add in some other key important features. So let me back up. Myosin head, purple, up top, thick filament. The only protein that makes up the thick filament is myosin for the purposes of our class. We'll just say that's it. It's about 99% myosin. Each sarcomere contains about 200 of those myosin heads. Every single sarcomere, there's 200 myosin heads. So there's 200 molecules of ATP per sarcomere. That's a lot of energy. Down at the thin filament, 3 proteins to remember. Actin, also referred to as G-actin. It's colored in red. It's the round spherical balls that make up a majority of the thin filament, but you have the cylinder tropomyosin proteins that hide or cover the binding site for the myosin head. You haven't told us, Keller, what troponin does. You just said it's a regulatory protein. I'm going to tell you here in a second. I just want to make sure we're going one step at a time. Everybody follow that? Any questions on that so far? Okay. So if we fill this out, We categorize myosin and actin as contractile proteins, and then we would actually refer to tropomyosin and troponin, and I already said this, as regulatory proteins. They regulate. They're the gatekeeper. They determine whether or not these contractile proteins see each other. Now I need to tell you how they do that, right? I need to explain to you how they do that. But before we go there, and we'll get there slowly, and we'll cover that in more detail on Monday, I want to talk a little bit more about the architecture. Okay, so I want to introduce this kind of slowly. And at the end of the lecture, we might get to more of the mechanism. And some of you may be like, oh my gosh, that was so much information at the end. Okay, it's so nice outside. I was thinking about the birds chirping, and I should have worn my flip-flops, and I don't remember what you said at the end. We'll start on Monday where I end, so we have it again. I have office hours tomorrow, noon to 1. I have office hours on Wednesday, 11 to noon. And I have office hours on Thursday. Don't remember the Thursday ones. Hang on. Thursday's so far away from me right now. Hang on. What did I say? Does anybody remember what I said? 1:30 to 2:30 on Thursday. 11 to noon on Wednesday. 12 to 1 on Tuesday. Wednesday I'll be in person in my office in Weta. All of them I'll be on Zoom. Okay? So let's continue with the microanatomy. You following? We're back here. Let's talk about the sarcoplasmic reticulum, the terminal cisternae, and these T-tubules, the triads. There is a lot of nomenclature in this unit. And if you can remember this nomenclature, and you can remember what it does and put the story together, then excitation, contraction, coupling that Matthew's excited to learn about will be easy. Okay? So back to the sarcoplasmic reticulum. This is a modified endoplasmic reticulum. It's similar words, right? Sarcoplasmic reticulum, endoplasmic reticulum. If you remember what the endoplasmic reticulum did inside the normal cell, is it packaged things and put a membrane around certain things. Here we're packaging calcium. So the role of the sarcoplasmic reticulum in a muscle cell is to store calcium. So that the calcium is inside the cell but in a warehouse, like a silo, a bag of calcium. So it's not in the cell free floating around. And you'll see why that's important. The terminal cisternae, that is the end portion of the sarcoplasmic reticulum. Remember that I said the sarcoplasmic reticulum is like this sort of like spider network right here. In this tubes are connected to this larger blue tube, and then there's a purple tube in the middle. There's 3 tubes here. That's the triad. 2 on either side are the T-tubules. No, sorry, the terminal cisternae. And the purple one down the middle is the T-tubule, transverse tubule. And then there's more network of sarcoplasmic reticulum, more network of... It's everywhere. It's all throughout the cell. The triad consists of this architecture. A T-tubule down the middle and 2 terminal cisternae on either side. The T-tubule down the middle, I'm going to tell you right now, give you some foreshadowing to Matthew's question. That's where the action potential goes down, is the T-tubule. So it comes from the nerve, it crosses the synapse, it goes onto the membrane of the muscle cell, which is referred to as the sarcolemma. It runs across the surface of the sarcolemma down a T-tubule or a transverse tubule into the inside middle part of the cell. On either side of it are bags of calcium, and that's the terminal cisternae. And that electrical action potential that goes down that T-tubule is going to trigger calcium gates to open and allow calcium to leave the sarcoplasmic reticulum, specifically the terminal cisternae of the sarcoplasmic reticulum, and into the cytoplasm of the muscle cell. Did you follow that? You want me to say it one more time? Okay. Nerve physiology. Action potential comes down the axolemma, which is just the membrane of the cell. It goes to the end of the synaptic knob. Now acetylcholine crosses that synapse. It binds on the other side. We're going to add in some more details. It propagates the action potential now down, instead of a neuron, down a muscle membrane. The muscle membrane, instead of a axolemma, is called a sarcolemma. You see the change in nomenclature? Down the sarcolemma, it moves deep into the muscle, down what's called the transverse tubule, which is the middle of the triad. That transverse tubule takes that electrical signal into the middle of the cell. The whole time it's moving, on either side of it is a specialized part of the sarcoplasmic reticulum called the terminal cisternae, which store calcium. And that electrical signal is seen by a calcium channel that will open and allow calcium to move not outside of the cell, but from inside of the cell in a storage house to the cytoplasm of the cell, to be available to find the troponin complex. And that's what troponin does. It's going to bind calcium. Okay? We're going to do this a few more times. Don't worry. Yep.
Does it hit just the first triad, or does it hit them all?
Oh, that's a great question. Does the action potential just hit the first triad, or does it hit all of them? If the signal is big enough, it'll go down and it'll propagate. So it'll go down, come back up and go. Down, come back up and go down. Right? Does that make sense? So it comes down, out, down, out, down, out. Much faster than I just drew. Okay? You tracking so far? Kinda? Sort of? Okay. So back to the myofilament. So pause that concept. Let's talk about what we're linking these things to. So here is our architecture of our thick and our thin filament. Here's our Z-disc to orient you. So we're on the left side of the sarcomere, and the other part of the sarcomere is somewhere over here on the wall. And you can see this protein that's very defined that links the thin filament to these other linking proteins is dystrophin. And these linking proteins ultimately link this outside of the cell to the extracellular matrix that anchors the myofilaments, and it helps to align them for optimal contractile effectiveness. And there's many of these molecules, 7+ proteins that are linking proteins, and we're going to really just hone in on one, which is dystrophin. And this dystrophin is really the most clinically important because it transfers all the force immediately to the other linking proteins. And unfortunately, there's this huge genetic disorder of this dystrophin protein, or the gene that codes for this dystrophin molecule, that produces a very debilitating disease known as muscular dystrophy. So in the category of muscular dystrophy, there's a whole group of these heritable diseases where the skeletal muscles degenerate and weaken, and they're replaced with scar tissue and fat, which is non-functional in the case of a muscle. In the case of Duchenne muscular dystrophy, it is a sex-linked recessive trait, so 1 out of 3,600 live-born males are affected. It's the most common form of the family of muscular dystrophy diseases. It can affect females, it's although rare. It usually is diagnosed at an early age, 2 to 10 years of age. The mutations in that gene that codes for the dystrophin molecule. And essentially what happens is you can contract your muscle cell, you just can't link it to the outside of the muscle cell, so it doesn't do anything. It's kind of like that tire on ice that's just slipping. and it causes damage to the tissue, and that's why you get scar tissue, which is part of a healing response. Unfortunately, the life expectancy is low because cardiac and respiratory muscles utilize this dystrophin protein. Okay, so cardiac and respiratory distress are problematic. As of now, there aren't any cures. There are treatments to try to minimize it, but there aren't any cures. Questions? Questions so far? All right. The neuromuscular junction. So we're building up to go back to that story mechanistically of how contraction takes place. And I'm giving you snapshots, kind of foreshadowing what we're going to talk about. And those are the last slides of the lecture. But I'm trying to build up the story on how the structure leads to the function. So the structure we need to talk about next is the neuromuscular junction, NMJ. And if you remember the name, you should be able to figure out what it is. It's where the nerve and the muscle meet. So instead of nerve, synapse, nerve, it's nerve, synapse, muscle. Neuromuscular junction. So this statement is really important because if you denervate a muscle, a skeletal muscle, you cut the nerve, it won't contract. It needs a nerve signal to do something. The nerve connections can be severed, they can be poisoned. You denervate a muscle and you're going to have atrophy almost instantaneously. Okay? So if we have patients that have spinal cord injury and they have paralysis of the lower limbs, they will atrophy their leg muscles very fast. Now, the future physical therapist in the room, you can do active contractions. Like you can, or passively move and contract their muscles to try to minimize the amount of atrophy. But it's not nearly as efficient or effective as us contracting our muscles on our own. And that includes postural muscles too. So this happens not even in a denervated state, but it happens in patients that are casted or they're wheelchair bound or they're bedridden, they're on bed rest. They'll lose muscle mass significant very quickly because it's not being activated. Now, what's fascinating to me is when you look at some of the motor neuron arrangements where we've got motor neurons and motor units, we've got somatic motor neurons where we've got a nerve cell whose cell bodies are in the brainstem and spinal cord that serve the skeletal muscles. We've got somatic motor fibers where they're axons that lead to the skeletal muscle. Each nerve fiber branches out to a number of muscle fibers, and each fiber is supplied by its own motor neuron. So let me show you a picture to describe this. So if we look at a familiar picture up top in the right, we've got a spinal cord slice. We see that our anterior or our ventral side that's in the front is where our motor efferent with an E pathways are coming out. We have one motor neuron that's up top, colored in blue, that's going to go to its corresponding fibers. We have a motor neuron down low, motor neuron 2, that's colored red, that's going to its corresponding fibers. So the organization is the motor unit is the nerve fiber and all the muscles that's innervating. All the muscles that one nerve fiber innervates is considered the motor unit. Now, you can organize these a different way, in many different ways, to get different types of results. So we've got muscle fibers of one motor unit that are dispersed throughout the entire muscle, like what you see here. Here we've got two motor units. Make sense? we've got two motor units because we've got one motor neuron that is innervating half of the fibers, another motor neuron 2, that's innervating the other half. But they contract in unison because they're getting the same signal at the level of the ventral root horn as it leaves the spinal cord. So you can produce a contraction over a wide area, a weak contraction in that example. Okay. So this provides the ability to sustain contractions for long periods of time, like sitting or standing, postural muscles. If we want effective contraction to last, then we typically have to recruit several motor units all at the same time. Make sense? Now, let's talk about some of the details of the average motor unit is about 200 muscle fibers for each motor unit. That's on average. Some are bigger than that, some are smaller than that. If they're smaller than that, it's for fine control. So 3 to 6 muscle fibers per neuron for like hand muscles and eye movements. Now, the opposite strategy is true. You want more strength over control, like the quadriceps or the hamstrings, biceps brachii, triceps, large motor units, powerful contractions supplied by a large motor unit. So the example here is the gastrocnemius, down in the calf of the leg. 1,000 muscle fibers per neuron. But you're not going to take notes with your gastrocnemius or your soleus muscle, are you? You with me? You need powerful contractions. Many muscle fibers per motor unit for explosive contractile capability. So the NMJ, there's a synapse where that nerve and that fiber meet. The target cell here is actually a muscle cell or a fiber, muscle fiber, rather than a nerve. Each branch, terminal branch of the nerve fiber forms a separate synapse with the muscle fiber. And the myelin sheath actually encases that synapse. It keeps it enclosed. So the neurotransmitter that's released is actually housed within the synapse. It doesn't just leak out. One nerve fiber stimulates the muscle fiber at several points within the neuromuscular junction. It's not just in one location. So this is a picture of the NMJ, the neuromuscular junction. So let's orient ourselves. We've got the sarcolemma up top. What is the sarcolemma? Who can define what it is for me? It's the plasma membrane. It's a phospholipid bilayer. Right? It's got hydrophobic tails, hydrophilic heads, right? It's got transmembrane proteins. It's the fluid mosaic model. It is the membrane of the muscle cell. Sarcolemma. Why is it sarcolemma? Because sarcomere, remember, that's like a unit of flesh. So that's why we call it that. Here at the synapse, hopefully you can appreciate that the sarcolemma has these folds. Why do we see these folds? Anybody have any ideas why that architecture exists like that? Folds. And I'm going to go in the back, okay, get some new blood. It's to increase surface area. I love it. You've heard that question before, maybe. And look at all of these inserted receptors. These are acetylcholine receptors on the other side of the gap. These spheres are synaptic vesicles at the synaptic knob. This is the end of the nerve. This is where we used to be. This acetylcholine vesicle is going to fuse an exocytose, another unit 1 term, spit out the acetylcholine into the cleft, and it's going to find its acetylcholine receptor on the other side. Here, you can see the Schwann cell. We know about the Schwann cell up top. Here's the Schwann cell here that actually forms this covering. Okay, it's like, all of my patio furniture has been covered all winter, and now I'm pulling the cover off. So it's protecting it. It's keeping all of this acetylcholine in this location to maximize the opportunity. It's going to go to the other side, and it's not just a single membrane. It has all these folds, so you can add in every single one of these rows of acetylcholine receptor wouldn't exist if this was just straight. Can you appreciate that? So you're maximizing your opportunity to couple that signal to the other side. What's down here? What's this, all this stuff? Just tell me generally, what is it? Where am I? Where am I? Inside the muscle. This is inside the muscle. How do you know? Well, here's the nucleus. Here's another nucleus. Remember, skeletal muscle is multinucleated. Oh, look, there's lots of mitochondria as well. And, we just have intracellular protein, actin and myosin, but we're in a muscle cell, so it's organized in a very specific pattern. You following? Okay. So the synaptic knob, that's the swollen end of the nerve fiber. Those acetylcholine neurotransmitters are packaged there. They're going to cross the cleft, they're going to bind, they're going to excite and have an excitatory postsynaptic potential. And I haven't told you exactly how that's all working because I want to build up the story slowly so you can understand where all these parts fit in. The cleft, tiny gap between the knob and the muscle sarcolemma. Schwann cell envelops it, isolates the neuromuscular junction from the surrounding tissue fluid. You could probably anticipate if you don't have that Schwann cell protection, you release the acetylcholine. If it's a fluid environment, you're going to lose a lot of that chemical messenger just leaking out of that cleft. You want to maximize and protect your signal because there's a lot of biological energy making acetylcholine, putting it in a vesicle, spitting it out into the gap so it binds on the other side. The synaptic vesicles, exocytose, we talked about that. We have over 50 million acetylcholine receptors in the body that are incorporated into the plasma membrane of a muscle cell, which is also referred to as the sarcolemma. Those folds are called junctional folds. That's these right here. Junctional folds. That's the formal term for them. Nailed it in the back, increased surface area. Maybe you were reading ahead, which I love. If you have a lack of receptors, this leads to a decoupling of excitation-contraction coupling, and this is what happens in the disease known as myasthenia gravis, which in the Latin refers to as muscle weakness. So you can see all sorts of different places clinically where something could go wrong. And you all know that I like to ask those types of questions on the exam. Like, hey, what would happen in this patient if there was a depletion of acetylcholine receptors along the sarcolemma and within the junctional folds? Which of the following might happen? And I have A, B, C, and D. Well, if there's not enough acetylcholine receptor, A, you're going to accelerate the speed of excitation-contraction coupling. Yes or no? No. You are going to increase the amount of acetylcholine that binds on the postsynaptic side. No. You'll decrease it because if you don't have enough receptors, you can release all the acetylcholine you want, right? You're going to eliminate the ability to propagate excitation-contraction coupling. I would agree with that one. So this is how I write exam questions. That's why it's important for you to come to lecture or listen to lecture and try to understand, okay, where would Dr. Keller ask questions about how this works, and how would he ask those questions? Because to be honest, those are the kinds of things you'll remember. Next fall, 2 years from now, you may not know all the details. You can Google those details, but you'll remember how it all fits together if you can remember some of these things. The basal lamina. This is a thin layer of collagen and glycoprotein that separates the Schwann cells and the muscle cell from the surrounding tissue. And it contains, importantly, acetylcholine esterase, abbreviated ACHE. And this breaks down acetylcholine after contraction, causing relaxation. Because you don't want acetylcholine bound to its receptor infinitely. Bless you. Okay, you would have what we call spastic paralysis. You'd have muscle contraction that would be frozen in time. So let's look at just a couple of different pictures. I really like this figure because I think it clearly describes microanatomy of where we are, and it puts the nerve unit together with the muscle unit. Oh, questions about Tourette's? Let's talk after lecture. How's that? Because I want to try to get to a point where we can hit the pause button. And then we've got office hours tomorrow and Wednesday before lecture on Wednesday. And Wednesday we're going to get into, today's Monday, right? Yeah, Wednesday we're going to get into more of the excitation, contraction, coupling. So this is a pretty cool picture. Look, here's 100 microns. You can see individual muscle fibers. This is the histology, if you remember, back in lab and in lecture when we looked at striated muscle. You can see the striations. That's the light, dark, alternating pattern of the thick and the thin filaments. You can hopefully appreciate, it's labeled the motor nerve fibers. They're coming down. And then they terminate here at a neuromuscular junction. Why do you think there's all these dark circles? What do you think all those dark circles are? Any ideas? I don't see any. Yeah. Acetylcholine or their receptors. Could be either one. It's hard to discriminate. But they're definitely acetylcholine, either the vesicles that house them or the receptors on the other side. So this picture, I think this is pretty cool. You can kind of see how it targets the muscle fiber with this individual nerve. I mean, this is the wiring that happens in your body to allow you to sit there attentively, take notes, allows me to stand for an hour and 15 minutes, allows you to run, jump, right? High five, play your sports, play your musical instruments. Here's the skeletal muscle fiber highlighted in blue. Axons of the motor nerves that you can kind of see they're strategically moving to their targets. This one's highlighting the motor end plate. The motor end plate is the term of the postsynaptic side. It's called the motor end plate. Let me say that again. The motor end plate is a term that refers to the postsynaptic side of the neuromuscular junction. Some more SEM pictures. This is really cool, in my opinion, this electron micrograph, where here you can see the cleft. Here you can see the synaptic vesicles. Highlighted here, and they're in dark, you can actually appreciate these are the mitochondria. And here's the junctional folds. Here we've got the skeletal muscle fiber, which would be referred to as the motor end plate, right? The other side. Primary synaptic cleft, our junctional folds, just another view. Synaptic vesicles on the presynapse side, and the postsynapse side is the motor end plate. Mitochondria, lots of mitochondria for a lot of energy, because you need a lot of energy to make this work. Okay. Let's talk about paralysis. We're not going to get into the last few slides today. It's okay. I have time to start it, but I won't have time to finish it, and I'd rather start there on Wednesday. So let's talk about paralysis, and we'll get out just a couple minutes early. Toxins that interfere with synapse function and the muscles can lead to paralysis. So we've got some pesticides and cholinesterase inhibitors. They'll either bind to acetylcholine esterase and prevent it from degrading, right? And now you get what we call spastic paralysis, a state of continual contraction of the muscle. These poisons lead to suffocation. And some insects can do this. They can make a poison that will transmit as a sting or as a bite, as venom, and cause spastic paralysis. Tetanus, lockjaw. This is formed from a toxin, Clostridium tetani, different type of spastic paralysis. Glycine in the spinal cord stops motor neurons from producing unwanted contractions. And this tetanus toxin blocks that glycine release. Causes an overstimulation of the spastic paralysis of the muscle. And lastly, we look at flaccid paralysis, and then we'll come back to this Botox example. Flaccid paralysis, in contrast to spastic paralysis, instead of contracted, it's relaxed. That's what the flaccid means, is it's not contracted. So the muscles are in a limp state, and curare in parts of South America have been harvested and coated on darts of certain tribes to cause paralysis of their prey, whether they're hunting or whether they're actually defending their tribe. And the curare poison will leave the muscle in a non-contractile state. And then we looked at Clostridium botulinum, either from food poisoning that can be excessive, or in our case, earlier in the semester, we looked at using this Clostridium botulinum cosmetically to eliminate wrinkles. Now think about this. Now you understand mechanistically how this works, right? If you block the release of acetylcholine, you cause flaccid paralysis, the muscles won't contract. Those muscles in the face that lead to the crow's feet and all the grooves in my forehead don't contract and go smooth. And companies have actually leveraged that for wrinkle removal. And you remember back when we looked at that, it's temporary because the mechanism here is it cleaves the docking protein for the acetylcholine vesicle to fuse. And so the cell just manufactures a new docking protein. Just takes a couple of months, and now you have plenty of SNAP-25, and you got to go back for another injection. Okay, it's good for the med spa. We're going to pause here. We're going to pick it up on sliding filament model on Wednesday. Okay? Have a great evening. Sun should be out for a little longer. Enjoy.